Untitled, so I'm going to talk about how plants tell time, and I hope I can convince you that that is important, so why that matters. And yeah, I can keep on going doing jokes about time. We are running out of time, and I need to be in time, but the good question is what is time? And this is a very difficult concept to define. Some physicists think that time is an illusion, doesn't even exist, so this talk is not happening. So it's a hot and controversial topic, and actually it doesn't really matter whether it exists or not. It's a useful concept. So what do we use it for? So we use it to measure change and to measure, to compare change or motion. So how many times my heart beats per rotation of the Earth around its axis. And so you're asking, so why that would be important? Why do you care how many times your heart beats? So it's not that I care about that, but it's an idea that it's a useful thing to have a standard unit to measure time. So time can be like a currency. This is from a Scientific American article in which they compare time to currency. So currency is what we use to compare value of a car with value of some shoes. And so you don't have to compare directly the values of the car to the shoes to something else. You just use some common thing to measure, a standard like the meter for measuring length. So we use time to measure motion change. And why is that important? Basically because we are living in a universe where things are moving, and most of them, they have periodic movement that they repeat. They have a cycle. And so we use clocks to measure changes on a daily basis, and we use calendars to measure changes on a yearly basis. And why that matters? So we heard about the beginning of this talk that there is something called the Argentinian time and the German time, and the Argentinian time goes a little bit slower than the German time, but we need something to put those together. We need to synchronize those, otherwise this meeting would not have started at the right time. And why do we need synchrony? And this is the key concept of my talk. So we need synchrony to survive. So we live in a changing world, and if we don't synchronize our cycles with the external cycles, we are dead. And this is a famous saying in Spanish. I don't know if Germany exists, but if you have a crocodile that falls asleep, he becomes the thing on the right side. So that's the key concept here. Synchronization is essential for survival. And that's also important for plants and has been an important issue that has driven even the concept of having a calendar for humans. So why did humans need a calendar? Just to synchronize their crops to the year, so they knew when to sow their seeds, so the seeds were synchronized with the changes in the environment. So it's measuring day with a clock is relatively easy. It was not that easy, but easier than a calendar. A calendar is something more complex, but organisms evolved a mechanism to measure the seasons quite effectively before humans conceptualized that. So, and the thing is that they measure, basically they measure day length. So as you move away from the equator, day length increases, and you know that the spring is coming and summer is coming, and the day length starts decreasing, you know, the autumn and the winter. And what do you need to measure day length? You need just two things. You need photoreceptors to measure light, discriminate light from darkness, and you need a clock. You need something to measure the passage of time. You need to integrate those, and if you do it the right way, you can tell whether you are in the spring, or maybe you can need to combine that with temperature, because day length is the same in springtime and at the beginning of spring and autumn. So you need low temperatures combined with longer days, then you know you are in. You pass the winter, you are getting into the spring. So plants have a mechanisms to do that, and they use it to time key biological processes throughout the year, so they know when to flower. Some plants prefer to flower at the end of the spring. You have wheat, peas. Some others prefer to flower at the end of the summer, or at the beginning of the summer, but you harvest them at the end of summer, beginning of autumn, like soybean and maize, and that's very important for our country. So it was like, 10 years ago, I could contribute something when I was doing my postdoc to understand the mechanism behind seasonal time measurement or day length measurement. And actually, I provided some data supporting the hypothesis of a German scientist, Erwin Benning, in this 1936. And he proposed that organisms measure day length by using an internal clock that controls the level of some substance. Could be here as the expression of a gene called constans. And that substance, the activity or levels were also light dependent. So here you have a messenger RNA that cycles and produces a protein. The protein is unstable in the dark, so if you don't have light, you only have high levels of the precursor at the time of the messenger, at the time when there is no light, so the protein will get degraded. If you increase day length, then at the afternoon, you will have a lot of precursor messenger RNA at a time where light perceived by photoreceptors, cryptochromes and phytochromes, will stabilize the protein, and then you will accumulate the protein, which is a transcription factor, which promotes the expression of a gene that Detlef Weigel showed more than 12 years ago, that it's a key protein controlling flowering time. So whenever there is a coincidence between an internal cycle controlled by the clock at, let's say, 6 p.m., so the plant looks at her clock, it's 6 p.m. Is there light or is it darkness? So it says, well, there's light because my photoreceptors tell me that. So constant messenger RNA levels are high, so there is protein being synthesized, protein being stabilized, and then the protein does its job, produces FT, and the plant flowers. So this is more or less a bit more complicated now, but the basic things still holds true. So plants still light with photoreceptors, I mentioned, phytochromes, cryptochromes, blue light, red light, and if you mutate them, you can have blind plants. And that's the way. So how they measure time now? So I explained barely or a little bit superficially how they measure the year or the seasons, how they measure time. What's their clock? And basically, clocks are all over living organisms, even in bacteria. Maybe some bacteria don't have it, but some do have. The rest of the organisms, they have a clock that a daily clock allows them to time processes throughout the day. And they are based on an oscillator made of some feedback loop, negative feedback loop. Usually, it's thought to be a transcriptional, translational feedback loop. I will show you some data that I think there are other layers of regulation, and those loops are connected to input cycles and to output pathways through which they control many different processes in plants, like almost probably every metabolic thing. We need to ask Lothar, Bill Mitchell, and Mark Steele whether that is true or just an overstatement, but probably many metabolic pathways, physiological process, photosynthesis, closing and opening of stomata, and leaf movement, which is over there, and eventually flowering time, as I explained. So what are they? How are they made? Well, they are made on transcriptional, translational feedback loops, and this is an old model. I was lucky to contribute to that. I arrived to Steve K lab, which is probably the leading lab in the clock biology field in plants, but also in other systems. And when I arrived, they had identified the first clock genes, and I helped other guys to try to understand how that works. So what we figured out is that there are two pairs of genes, LHY, CCA1, which are single MIB domain transcription factors that they bind to the promoter of a gene called TOC1, a pseudo-response regulator. They repress its expression. CCA1 and LHY are highly expressed in the morning, and TOC1, therefore, is repressed at that time. And TOC1 somehow indirectly probably promoted the expression of CCA1 and LHY. So there was, at some point, if you don't get TOC1, you don't get to have, again, CCA1, LHY, those levels go down, and TOC1 starts to increase and promote expression of CCA1, LHY, the cycle starts again. So that's what the data at that time said. Things are a bit more complicated, probably a bit more, and we saw this picture early today. So the model is now the central, what we think the central oscillator are a bunch of interloop loops. And what we saw are, so there are some things that still hold 10 years apart, like CCA1 HY repressed TOC1 expression. Apparently TOC1 that is not a promoter of CCA1 HY expression, but a repressor, so things have changed completely, and that was this year that happened, a few weeks ago. A couple of papers in Science, PNAS, and Molecular Systems Biology. Is completely counterintuitive, based on data, but not on some part of the data, not other, but the modeling works fine and explains a lot of things that we didn't understand now. So what we do in the lab is what I'm going to tell you in the last 10 minutes very quickly. It's just to show you some examples of what we do. We try to identify new clock genes because we think it's important to find every possible relevant clock gene, not just anything relevant. And because those contribute to regulate flowering time, and flowering time is important for synchronizing production with the environment and eventually to provide us more food and biofuel. So this is what we do, and we use two different approaches. One is reverse genetics, the other is forward genetics. So reverse genetic means you do a bunch of microarray experiments, gets a lot of data, and try to make some interpretations and look for things that are probably going to give you some new candidates to test whether they're involved in clock regulation. So the clock is controlled by light. So if you want to understand how light controls the clock, maybe you think one hypothesis is that it's through transcriptional regulation. And so you can do a simple experiment, just give light to a plant that was growing in light-dark cycles, goes to complete darkness, and you can, but the plant has her own or his own clock, and it knows when it's subjective day or subjective night. You can give a light in the middle of the day or in the middle of the night and see what's happening. So the clock itself is sensitive to light mainly in the night, not during the day. So during the day it's completely insensitive. So most of the genes that are turned on in the plant by light will go up in the middle of the day. So if you look only at those genes that are mainly upregulated by light in the middle of the night, and then integrate that data sets with those that are clock regulated most of their time, you end up only with 12 genes compared to thousands if you look at those that are upregulated during the middle of the day. And it's not that they are not regulated during the day, it's that they are more upregulated by light in the night than during the day. So we find two genes, CCA1, gigantea, which we knew that they are controlling the clock, and we got a set of other genes which we call link, so light induced at night and clock regulated genes. And when you reverse genetics, when you have candidates, what you do is just go get the mutants, see the phenotypes, and that's what we did. So link 1 doesn't have a phenotype when it's a single mutant. Link 2 has a long period phenotype, and when you remove link 1 and link 2, which are homologs, you completely screw the clock and you get an arrhythmic plan. And if you don't have a clock, you don't measure the calendar. The calendar is not working, so you don't set the time precisely. So we are figuring out how they work within this this network, but this is an example of what we do, and we are trying to place this in the context of other clock genes. The other is forward genetics. So we just go ahead and mutagenize plants and try to get mutants that their clocks are not running properly, and then try to see what their genes were affected. So we identified this, link 1, so we are not very, I'm particularly not very clever thinking different names, so light signaling and clock defective 1. So this mutation affects both flowering time and the clock. The clock is it's a slow clock. It has a 26, 27-hour period. So when you identify the gene, which we did by positional cloning, we found that it causes an enzyme called PRMT5, protein arginine methyltransferase 5. And what this enzyme does is transfer methyl groups, like we listen in the epigenetic talk, but instead to lysine, it transfers methyl groups to arginines. And it has many different targets. So it can target histones and epigenetically control gene expression, or it can target RNA binding or RNA processing proteins and regulate something in the life of the RNA molecule. And we didn't have a good signals that this was acting epigenetically. Instead, we had signals that this was acting through RNA processing. Actually, we did some microarray experiments, and we also had a friend working with flies. So the gene is conserved from yeast to humans. And there was a mutant in flies, and we analyzed circadian rhythms in fly, and they were screwed. Not the same way in plants. They were arrhythmic, at least behaviorally. which is what you see there is the activity. The black bar means the flies are active. And on the left, you see black and white because they are active in the morning and in the evening, and they have siesta and they sleep at night. In the mutant, they are screwed. So we use Affymetrix arrays in both to analyze gene expression and also to analyze RNA processing. And what we found in both cases is that it was affecting a subset of genes, particularly the RNA processing level. So what's the connection between PRMT5 and RNA processing? So one of the targets of PRMT5 in many organisms are core spliceosomal proteins. So the spliceosome is the ribonucleoprotein complex. It's a big ribonucleoprotein complex, actually several complexes working together to remove some parts of the genes. When the gene is expressed, not every part of the gene ends up in the mature messenger RNA that gets translated. So there are some parts that need to be removed, and this is done by the spliceosome, and there are these SM proteins, D1, D2, D, F, E, G. Those are part of the core spliceosome, and those proteins are targets of PRMT5. And PRMT5 acts with other proteins to target those, to methylate those. Nobody knew exactly what that methylation was needed for, but some people thought that that was necessary for spliceosome assembly. So what we found using genome-wide analysis, using gene expression arrays, is that this is an example of one gene. So you see the black bars are the exons, the thin lines are the introns. You see a gene is normally, when there is a white black bar, there is high expression. The thin line, the intron is being removed. But at one particular intron, you compare the wild type and the mutant, there is a difference. So there is an intron retention event in that particular intron, not in other ones. So out of 67,000 introns, there were only a few events that were affected in the mutant. And this indicates that the mutant was not affecting splicing everything, just a few things. One of the things that was affected was a clock gene, PR9, which has different types of splicing events. This was completely screwed in the mutant. PR9 is one of the core clock genes, a homolog of TOC1. In the fly, it happened the same. There is a core clock gene called period, has a splicing. So the top panel is the gene expression, which is a little bit twisted, but not dramatically affected. The bottom panel is the splicing of the per gene is completely disrupted. And what we saw actually is that among the events that were affected, the mutation affected, rather than constitutive splicing, it affected a subset of splicing events called alternative splicing, which was in the slide that I just skipped. So when you splice introns, you can splice in different combinations and you get different proteins or different sorts of outputs. So some introns are constitutively spliced, some they can go different choices, and the mutation affected mainly alternative splicing, and particularly affected splicing events in which one of the sites that is required to recognize the intron is not consensus site. So what we are doing now, and these sites are recognized by the spliceosome machinery through complementary between the RNA part of the spliceosome and the RNA part of the messenger RNA by complementarity. When what we saw is that the mutation affected events that were far away from the complementarity. So we think that methylation is important to stabilize weak interactions, and we are going now for kind of testing that idea using natural genetic variation, which is a kind of genome-wide mutagenic approach, and combining that with RNA-seq in collaboration with Delef Weigel. So I'm running out of time, so I'm going to skip some of the slides I have and just go to finish with a summary of what my point here was. Basically, we identify